Wirth from the Peter Doherty Institute in Australia. I'm going to talk about our tool Barbet that we developed for bacterial taxonomic classification. So microbial genome classification is a resource-intensive process typically. There are some fantastic resources out there for doing genomic classification, such as GTDB. However, GTDB, the latest version of the database, requires over 100 gigabytes of RAM to run and over 100 gigabytes of hard drive space. So we decided to train a deep learning model to perform the same task as GTDB, but in low resource settings. So we trained this model on over 700,000 genomes from 140,000 different species using the latest GTDB database. The model uses a protein language model internally to embed the genomic sequences and then a further deep learning model to build a hierarchical representation of these genome embeddings. Our model includes several different stages, much of which are similar to GTDB TK's classification system. We extract marker genes from assembled contigs and then embed those marker genes using a protein language model. Following the embedding, we trained a deep learning model to produce a hierarchical representation of the taxonomy and assign probabilities to each of those taxonomies. This tool is implemented as a command line interface as well as a web interface. Users can go to this web interface and drag and drop their samples on there to produce results. The outputs from our model are a probability distribution on all species within the GTDB taxonomy. You can then process this probabilistic model to identify the most probable taxonomic assignment. Here you can see an example of these probability assignments where we are 100% confident all the way up to the family, and then the genus and species have more diverse probabilities assigned to them, and so we can see that in the figure. We benchmarked this method on several different publicly available data sets, including 1,000 genome isolates from a public health lab and over 3,000 metagenome assembled genomes from a human gut microbiome. The model achieved significantly high accuracy. We found that in the public health data set, we were able to achieve 99% accuracy compared to GTDB TK, and in the human gut samples, we achieved 97% accuracy. For novel species, that is species that the model hasn't seen at all, we were able to achieve 95% accuracy at the genus level. The real power of this model is its ability to run in low resource settings. Our model is 98% smaller than the GTDB TK database, which enables you to run this on laptops. It's also significantly faster than running on a single isolate and can be GPU accelerated. In addition, we use significantly less memory than GTDB TK at around 2 gigs. So this is truly a tool that can be run on a laptop. So this work was the result of a collaboration with the Melbourne Data Analytics Platform. And I'd like to thank my collaborators, Robert, Ma, and Gabriele, for their contributions to this project. And as well, my advisor, Thorsten Seaman, for contributing his knowledge of species identification.